the honor today of introducing Dr. Sincan, Murat Sincan. I'll be brief. Dr. Sincan got his medical degree in Turkey just a little over 2 decades ago, I think, yes. Cross-trained at Harvard with a tremendous academic pedigree. Before departing the university where you received your medical degree in Turkey, I understand that you were chief information systems architect at that institution. So a lot of informatics horsepower early on. And then to kind of reduce a long CV to a few salient pieces, Dr. Sincan came to the States to pursue and pursued eventually additional postdoctoral training in DC and Rockville at NIH in the National Human Genomics Research Institute a little more than half a decade ago, and then immediately took a post there at NIH as a staff scientist. And then 4 years ago or so, we were lucky enough to recruit you here to Sanford and the Imagenetics Initiative and our Sanford School of Medicine, where Dr. Sincan holds a position as an assistant professor on the academic track here in internal medicine. Dr. Sincan's had many national and international contributions to the field of informatics. A few years back, he was a panelist on a committee that standardized research data elements for the prestigious American Medical Informatics Association. So he's truly an international leader in the field. Before I give you the podium and ask you to teach us, I'd also like to introduce one of Dr. Sincan's colleagues today who will be co-presenting, and that's Julia Stish. We're also delighted to have you here bringing extra horsepower, intellectual horsepower to the table and helping Dr. Sincan's program move forward. Julia is a medical student in the class of 2023 with a diverse interest, but I think a strong growing interest in pediatrics and the interface to maternal fetal medicine. And I think where I'm taking you here is that you're keenly interested in the use of genetic testing to improve risk stratification and screening for common diseases. So she'll be sharing some of her work with us today along with Dr. Sincan. So you're going to lead. Yeah. Julia's going to go. Yeah. Very good.
Hello, I'm Julia, and I'm just going to start the presentation for Dr. Sincan. We are talking about Phenome-wide association in the advancement of precision medicine. In order to talk about PHEWAS, we have to talk about our genome-wide association study first, also known as GWAS. This is a tool we use in genetics research to associate specific genetic variations with diseases. This involves scanning the genomes from many different patients and looking for genetic markers that can be used to predict a certain disease state. These genetic markers are then used to understand how genes contribute to the disease and help us develop better prevention and treatment strategies for our patients. In order to kind of understand GWAS and PHEWAS, we do need to go back to some basic genetics and talk about our single nucleotide polymorphisms or variations. We call those SNPs for short. These are basically places in the genome where people differ. They are substitutions of single nucleotides at a specific position in our genome. So most of these SNPs don't do much, but some do confer risk of diseases such as diabetes or heart disease. How do we test for these at Sanford? Here at Sanford, we use Illumina Global Screening Array. This is a genotyping array used for population scale genetics, variant screening, and precision medicine research. To date, there's been over 5.5 million samples run globally. Over 600,000 markers have been found, and out of that, 50,000 research variants have been curated, including markers for pharmacogenomics, newborn screening research, and risk profiling. So going back to GWAS, this is when we take our disease and then study the genotypes of our cases and our controls from a population. Once we find this disease and find the genetic marker, we perform regression modeling. Then, of course, we adjust for covariates such as age, sex, ethnicity, and then we correct our findings in order to get a p-score which is less than 5 times 10 to the negative 8. This is our genetic significance threshold. And of course, it's advised to replicate and validate such findings. So when we look, genetic variants go across a wide spectrum of effect size and allele frequency. What we're interested in when it comes to GWAS is our lower right quadrant. That is what we're interested in, common variants with small effects. If we go a little bit higher up to the left upper quadrant, that is when we are getting into our classic Mendelian diseases and such. So GWAS findings to date. As of this May, there's been over 5,000 GWAS have been published for, and over 250,000 SNP trait associations at that genome-wide significance threshold, that p-value less than 5 times 10 to the negative 8. In order to transition into talking about PheWAS now over GWAS is talking about pleiotropy. Pleiotropy is basically when a single gene affects multiple outcomes in a person. There's very abundant evidence for pleiotropy. Notably, we can talk about our human leukocyte antigen region, HLA region. This is a genetic risk factor for Graves' disease, type 1 diabetes, multiple sclerosis, lupus, and rheumatoid arthritis. So all of those diseases share the same genetic risk factor area on the genome. Now I'll have Dr. Sincan talk about a little bit more in depth about my introduction. So thank you.
Thanks, Julia. So thank you all for coming and joining us today. I'm actually very pleasantly surprised that there are no technical issues and we can smoothly transition between talks. So that was a great introduction, right? We need to understand genome-wide association before we can actually talk about the phenome-wide association, which we'll go into a little detail later. But to dig a little deeper into the pleiotropy, I want to share this slide with you. Here you see different types of pleiotropy that we can observe and possible ways that they can manifest in the genetics when we interrogate the genomes of individuals. In section A, which is the classic pleiotropy, where there's a causal SNP that's represented by this red star, is having a causal impact on two different phenotypes, P1 and P2. And we don't usually have the ability to detect these causal SNPs or causal variants through genotyping, for example, with the SNP chip that Julia just showed us. But we usually have these tag SNPs that are represented by these yellow circles with an S in it. So the tag SNP, because it's close in proximity to the causal variant, is what's called in linkage disequilibrium with that variant. Therefore, it gets inherited with that causal variant most of the time because of their close proximity and because the inheritance happens in blocks, what's called haploblocks. So by detecting those tag SNPs, we can detect the presence or absence of these causal variants, which then, if they are pleiotropic, will have an impact on two phenotypes like this. Here you see a different way that this can happen when the same tag SNP can tag actually two different causal variants. And here one might be having an impact on the folding of the protein, the other might be changing an acidic amino acid to a basic amino acid and changing the electrophysiological properties of the protein, but still having similar impacts or impacts on two different phenotypes from the same gene, yet tagged by the same tag SNP. Here on section C, you see two different SNPs tagged by two different tag SNPs. So when we interrogate the genome using a genotyping array, we will have two different SNPs found that is associated with phenotype 1, and then the second SNP will be associated with phenotype 2. The problem starts when we go to section D, where we have this causal variant down here, which is having an impact on an intermediate phenotype. This could be, for example, increased BMI, which then will have an impact on a secondary phenotype. Now, when you interrogate the phenome, you will see these always kind of comorbid situations going together, and one might be actually causing the other. So an increased BMI can have an effect on various types of metabolic disorders, cardiovascular disease, diabetes, yet it's actually that single SNP that's causing this more intermediate phenotype that is now being observed. And if we're not aware of these types of situations, then the interpretation actually can be fairly problematic. And of course, due to design issues or ascertainment bias, there could be other things that can go wrong, but I'm not going to go deeper in those because our focus right now is not those. So here you see another example of, for example, Crohn's disease, which is an autoimmune disorder primarily, sharing genetic variation between itself and various other phenotypes. So this FADS1, which is involved in the fatty acid metabolism, is involved in both HDL cholesterol trait, triglycerides, type 2 diabetes, and glycemic traits, as you can see over here, and also Crohn's disease. So when we run genotype-phenotype association tests, we do observe these associations, and that's actually how people originally started observing these pleiotropic effects when multiple GWAS studies ran in the older days. Right? GWAS predates PheWAS. Many GWAS studies started detecting the same variant that are associated with different types of diseases. When a second GWAS was run on a different disease, yet the same variant was detected, that was a clue to the research community that there might be something else going on in terms of pleiotropic impacts of these genes and mutations that are harbored in those genes. So I alluded a little bit into this, that we have known for a long time that multiple genetic variants are known to influence multiple unrelated traits. This became very evident, even before the GWAS era. Even Mendel observed this, and people studied it with different genetic research tools before even the GWAS was available as a tool to us at the population scale. But we also know that genetic variants, once they are detected, let's say for a single disease, let's say for type 2 diabetes, we run a GWAS study and detect a number of variants that are associated with type 2 diabetes. Then we can take each one of those variants and then scan the phenome and see what other phenotypes or traits this variant might be associated with. We also know that the genetic effects may be dependent on environmental exposures and life stages. So are the phenotypes, and it's always good to be able to understand exactly what is due to the genetic effect and what is due to the environmental and life stages that are observed in these patient populations. And of course, comorbidities is a fact of life, and we need to understand better these multiple genetic factors that contribute to the etiology of these comorbid conditions. So PheWAS is actually a tool that allows us to do this. Now, to wrap our heads around this a little bit more, this is a really good representation of the differences between, similarities and differences between GWAS and PheWAS. On the top row here, you see the GWAS paradigm. We usually take a disease, say type 2 diabetes, and we take a population and we first define cases and controls, those who have the disease and those who don't have the disease. Here, they're represented by green, those who have the cases are green, whites are control. Then we genotype this whole population, and then we look at all of these variants associated with this specific phenotype. In the end, we end up with a list of genetic variants that are associated with this disease. This looks more like, PCSK9, so maybe it was hyperlipidemia that we studied in this case. But then we can take this PCSK9 SNP, which is reference SNP RS 505151. This is just an ID number for a specific variant. We can take that and turn the design of the study completely, flip it 180 degrees and turn it around and say, okay, now we know that this variant is associated with this phenotype of interest, right? Let's say hyperlipidemia. What else is it associated with? So we then take, and this is only available when you have really robust genotype-phenotype databases, linked electronic medical records to these genotype databases. This is only available when you have that, and you can say now each card here represents a different phenotype, right? Or you can think of a disease or a trait, but each phenotype now, the blue phenotype, the red phenotype, the yellow phenotype, the green phenotype, then we start asking this question, what of these different phenotypes are associated with this specific SNP? And we end up with a list of phenotypes, traits, and diseases that are statistically significantly associated with this specific genetic marker. I said we need robust genotype-phenotype databases. UK Biobank is a prime example of this, where you see for approximately 500,000 people in the UK, they have managed to enroll these individuals into their study and did a really good job of genotyping these individuals. But on top of that, not only they linked their national health system medical record data to this database, but they also focused on patient-generated data, which is a really good source of additional information that is typically not available from the episodic centered medical record that we have, right? So patients come in with a specific problem, and it's a focused history-taking, focused evaluation, and the information typically is limited to that. We don't ask patients about their travel history necessarily when they come in with a sore arm, right, or a broken limb. But patients can answer a wealth of information if they're given the opportunity to answer these questions through surveys. And also, in addition to that, there's a wealth of information that can be collected from wearable devices, for example, about their lifestyle choices, sometimes even location information, but of course, steps and heart rates and sleep quality information that is really has a lot to say to us about a lot of chronic conditions these patients might be suffering from. So, and there's also imaging data. So you can see this 360 data collection around the patient is really critical for us to be able to do these association studies. But once we collect this information, especially across multiple institutions, sometimes even within the same institution across time, it is important to be able to standardize this data so that it can be analyzed. First, it's computable, right? If it's free text, we need to convert it to some type of computable concept array. But even if it's just diagnostic billing codes, like ICD codes, it really helps us if we could harmonize, standardize all that information into a single common data model, which then can be shared across institutions. We are lucky here at Sanford, for example, for having a unified electronic medical record system that is used across the enterprise, but not every institution is the same. Sometimes a children's hospital might be using a different medical record system, adult hospital, even few hospitals within the same adult healthcare system might be on different versions or different medical record systems. So just combining that data into a single computable resource is actually a great undertaking, but it's a critical tool and technology for us to be able to conduct these studies that help us advance our understanding of the biology behind. So what's the relationship between PHIWAS and precision medicine? I'm just going to read you the definition of precision medicine. Actually, it's personalized medicine per the NIH. They, I think, still haven't updated their hyperlink. But precision medicine is using an individual's genetic profile to guide decisions made in regard to the prevention, diagnosis, and treatment of disease. So it really encompasses the whole spectrum of what we do to improve health for our patients. But the cycle, as you all know, typically is, the first step is hypothesis generation or observation. and then hypothesis generation. Then we study, these hypotheses through testing, and we perform discovery. Then we take those scientific discoveries we made and apply them to clinical setting as tests. In this context, it would typically be a genetic test, for example. And then we apply that result to prevention and treatment of our patients. This is really happening right now, right? In cancer genetics, in pharmacogenomics, at Imagenetics, we are applying all this information to everyday primary care. The oncology team is doing the same. Our pharmacy team is doing the same. There's really a great use of genetics in the rare diseases. But with things like polygenic risk scores, it is now becoming increasingly useful and more commonly available because there's just so many more people that can benefit from an application of these tools to common diseases in the common disease area. And there are various methods that we can use. For hypothesis generation and discovery, we could use GWAS and PHEWAS, and of course whole exome and whole genome sequencing, even targeted sequencing and genotyping. For clinical testing, almost all of these tools, polygenic risk scores, whole exomes, whole genomes, targeted sequencing, and genotyping can be developed as a clinical test with given parameters and approved by CAP, CLIA, all that. And then they can be, the information we get from there can be applied to guiding an individual's medical decisions for an individual in regard to prevention, diagnosis, and treatment of disease. So it really fits into that whole cycle of generating knowledge and then applying that knowledge to improving the healthcare that we provide. We have to do two things to conduct either GWAS or PHEWAS, right? First thing, especially for PHEWAS, is the robust phenotyping that we need to engage in. And phenotyping can be done in two ways. One, we could use an existing medical record on the left-hand side here. And I apologize there's a little bit of cutting of the image, but it's still, most of it can be seen. So here in the clinical visit, the patient's EMR entry can be, different ICD codes, different lab results, and these all go through a process of electronic phenotyping. Or it could be a clinical trial, a research study where we ask certain questions to participants and measure certain things, including their height and weight, etc. And then after a phenotype adjudication step, really what happens is, in the end, we end up with a harmonized and portable phenotype that then can be applied in the standardized human phenotype Phenome database, and then combined with the genetic variants of interest. And then we're able to do either a GWAS or a PHEWAS on this population. So most of the time, if you look at the literature, the phenotyping at this point for a PHEWAS study is done using ICD billing codes. And the reason why that is the case is because they're available, right? That's the bane of our existence. We have to use these billing codes that are commonly available, but they're not necessarily always representative because sometimes they're used for justifying a test, or sometimes they might just be a running hypothesis on what the patient might have, like rheumatoid arthritis, and in the end they turn out not to have that, but it's still in their record, right? But at least we have to be cognizant of this difference. Before 2015, we used as a system, as a country, a different set of codes, 14,000 or so of these ICD-9 codes, and maybe the younger folks have never used them, but they were less representative. They were more broad in what they represented. But since 2015, October 2015, we switched as a country to using ICD-10 CM, which is clinical modification of ICD-10. And ICD-10 CM now has approximately 69,000 diagnostic codes and has a wealth of procedural codes as well. But even ICD-9 at its 14,000 or ICD-10 with its 69,000 codes is still too granular for being used in a PHEWAS study. So what we do is we typically combine these codes into what we call phe codes that are broader concepts that represent certain clinical phenomena, And then we use them for our association testing. And I'm going to go into a little bit of detail in the next slides. So this is an example of what that would look like in the ICD-10-CM. This is just showing one section, diseases of circulatory system, and down here are the different hierarchical codes. And if you just focus on that part of ischemic heart disease, you'll see that there are subcategories underneath that, right? And then if you just focus on that I-25 category, which is chronic ischemic heart disease, then you get this. Right? These are the leaf level. So we've gone through some major branches and minor branches, and now we're at the leaf level. So this is what we would get from the medical record of a patient. And many patients may have a combination of these, right? They might have coronary artery dissection, they might have coronary artery aneurysm. But in the end, they're all coronary artery aneurysm and dissections, and they're all under this chronic ischemic heart disease category. So you can imagine how we can combine these low-level ICD codes into higher-level concepts that represent a certain trait, a certain disease grouping that we're interested in. This also helps us find greater number of patients that share this phenotype trait that we're interested in, and it also helps us reduce the multiple testing burden, right? If we've figured out the phenotype piece, which is really challenging, but let's say we've done a good job of doing that, then we also have to have the genotypes. And I want to just focus on two aspects of the genotype information here. Genotypes are de-identified, which makes it really easy to work with them. Unlike the medical record, which really is sensitive information, we need to protect it, we need to process it behind our firewall. We can't really tap into external resources that easily with the medical record data. But with genotype data, we can upload it to the cloud, hire a company to help us with that. So it's easier in that sense to work with. But another advantage of genetic data is that it's fixed at birth. Unlike the medical record that changes from day to day, if you get the EMR data from yesterday, it will certainly be different than today's EMR data because more patients will be diagnosed with new things today. But we could sequence or genotype a patient at birth and sequence it throughout their lifetime. Especially, this is, of course, there are edge cases like somatic mutations in cancer, etc. But for the most part, for germline mutations, this will stand true. And we could use either SNP chips or whole genome sequencing to detect these genotypes. Of course, whole genome sequencing is more expensive, generates more data, but there are tools like low coverage, shallow whole genome sequencing that is comparable at the price point and data size that can give us similar information to SNP chips. To be honest, the main cost is quickly becoming personnel, data management, and computing. On average, one person's genotype for 100,000 positions through this SNP chip arrays can be obtained for less than $100. Similarly, low coverage whole genome sequence genotype data can be obtained at the research grade. There are companies out there that provide this service for $50 a person right now. But once we get the data, then we need to find a way to store it long term. We need to find to process it, compute with it. And that cost really adds up very quickly, especially over time. And that's really where the majority of the investment needs to happen at this point. So now that we've covered phenotypes and genotypes, what about different types of PheWAS? Before we go into another classification, I just want to cover this quickly. One can potentially run a PheWAS study across the whole phenome or run a PheWAS across a selected limited number of traits. An example of this would be if we detect, for example, a novel variant or even an old one for, say, depression. We might be interested in seeing what other mental health disorders this specific phenotype is related to or may have an association with. Similarly, typically this is done in broad disease areas such as metabolic diseases, endocrine diseases, immunological diseases. So once a genotype is detected for type 2 diabetes, we might want to see what other metabolic disorders or endocrine disorders this might be associated with. So that will be a targeted PheWAS, whereas the comprehensive one would be just agnostically running this against the whole phenome that we have. And genotype-phenotype PheWASs, especially SNP phenotype or SNP phenome PheWAS, is not the only type of PheWAS. You can imagine you really need to have cases and controls. And those cases could be those who have the genotype, and controls could be those who don't. But it could be different things. And the phenome doesn't have to be only ICD-10 codes, right? So people have done LabWAS, laboratory-wide association system, using laboratory values. People have done transcriptome PheWAS. So instead of a SNP detected from a genotyping array, you can take transcriptome expression levels for genes, and you can then classify them as high and low, or changed or not changed, and run a PheWAS against that. Another study here from 2019, CNV PheWAS, copy number variation PheWAS. There are different regions in the genome that have polymorphisms in the form of copy number variations. And one can certainly study the association between those and the phenome, and they actually have a big impact on various types of diseases, especially in mental health. Autism, for example, comes to mind when one thinks about clinically implicating or clinically relevant CNV variation. And it doesn't even have to be genetics, right? I'll just share you a study after these slides on COVID, where they've actually compared the phenome and looked at the associations across the phenome with COVID outcomes. Statistically speaking, it's not that complicated. We use standard tools that are not deep learning or machine learning. We really need to utilize hundreds of years of old tests like chi-squared for a simple case control comparison. Or if we want to control, and typically we want to do that for age and sex and other environmental factors that we might be interested in and have an impact on the comorbidity, we might just run a logistic regression, which will allow us to control for those covariates. But of course, like any other multiple testing, any type of GWAS or PheWAS needs to be then corrected for multiple testing, and that's typically done by Bonferroni correction. Now, the first slide I started talking about was the types of pleiotropy, and this really comes back because running a PheWAS, as you can see from the slides that I covered so far, once we have covered that genotype-phenotype database, once we have created that, right? Once we have our genotypes ascertained from birth and our phenotypes, we have a pipeline that can convert the EMR data to computable phenotypes. Wonderful. We can have a database and we can apply these fairly standard statistical techniques that don't really require a supercomputer necessarily and create these associations and find these associations. But the challenge really comes, how do we interpret them, right? Remember that pleiotropic case where the BMI was causing something else and they were comorbid situations? Well, how do you know if it's coming from the genetics or if it's a spurious pleiotropic association like that? It could very well be true pleiotropy. The same biological mechanism might be causing these two phenotypes that are comorbid. And the genetic allele that we're studying might be driving that biological change. But it could be just comorbidity, shared by the same biological mechanism, but the allele that we're interested in is only having an impact on one of the phenotypes. We're observing that association because it's just an association, right? I don't know how many of you are interested in this like association jokes, but we know, for example, that drownings are highly associated with what? Ice cream consumption, right? Well, no wonder, right? Both happen in the summer. But of course drownings are not caused, drownings and ice cream consumption are not caused by the same genetic change. But if we did that, if we looked at that association, we would find that a bunch of people with the same genetic variant, in the summer would suffer from those two. Ice cream consumption is not necessarily something you suffer from, but would have those two traits. So association is never causation. We always have to keep that in mind. And that really is the third section here, section C, where the environment is actually related to those two phenotypes, right? And the allele may still be connected to one of the phenotypes, but even it might not be the case. And the false phenotype distinction, where we might be thinking that there are two different distinct phenotypes in place here, and this could be our study design flaw, but they're indeed the different manifestations of the same phenotype. So it really is very challenging to interpret the findings from PheWAS, and of course there are tools that we can use that, first of all, we need to replicate our findings in an independent cohort, and then we, whenever we can, take these associations and try to elucidate the functional mechanisms behind them in the laboratory downstream. So here's the PheWAS study that I gave you a teaser before that is from the Michigan Medicine. It's very recent. It's in, of course, no talk right now should be done without any COVID slide, right? So here's my COVID section. So what they did was they took 5,698 patients who tested for COVID, not tested positive, tested for COVID. Of those, between March 10, 2020 and April 22, 2020, of those 5,600 some patients, 1,119 tested positive for COVID, right? So that's the cohort. Then they selected two control populations. One, 7,211 were randomly selected, and the other 13,351 controls were selected for matching race, age, and sex. Then they did the usual PheWAS, right? So they took the ICD codes from the electronic medical record for these patients, of course, making sure that the codes they studied were entered at least 14 days prior to their admission or to their positive test result for COVID. So these happened before. And combined them to 1,781 Phe codes, PheWAS traits. Then the analysis was performed after selecting for phenotypes. So of these 1,781 traits, some were quite rare and were not found in more than 10 COVID positive patients, so they had to be excluded from the study. Once they have achieved that, then they've done 4 analyses. 1, who got COVID versus who didn't. 2, who got admitted to the hospital. 3, who got admitted to the ICU. And finally, 4, who unfortunately ended up dying from COVID versus not. So this is just another summary of what they did from the 1,781 codes by selecting for those that are at least present in more than 10 individuals. They were able to find 1,043 Phe codes, and 778 patients had those. So of that, the breakdown with race, and this study is really about the differences between non-Hispanic whites and non-Hispanic African Americans and how their outcomes changed from a phenome-wide perspective. So 50% approximately were non-Hispanic whites, about 35% were non-Hispanic African Americans, and these are the breakdowns of who got hospitalized, who got admitted to the ICU, and how many ended up dying as part of this cohort. So I know this is a very busy slide, but I'm going to try to walk you through it, and hopefully it will make sense. So the top row is the full cohort. The middle row is the non-Hispanic whites, and the bottom row is non-Hispanic African Americans. The left-hand column is hospitalized versus non-hospitalized. So cases would be those who got hospitalized. Controls would be not hospitalized. The middle column is ICU versus non-ICU. And the right-hand column is deceased versus not deceased. Right? So we're not going to go over all of this, and this is called a Manhattan plot. And the reason why it's called a Manhattan plot, it's originally, from a GWAS world. It's because, they look like skyscrapers. So as they, get higher, so that's, I didn't name it, don't blame me. But these red lines, if you can see them, they are the genome-wide or phenome-wide, depending on the type of study you're running, significance levels. So anything above that has a p-value that is above that significance level that is controlled for multiple testing. And you can see there are some things that have crossed that line, that are significantly associated with whatever the outcome is studied in that box. Those of 1,700 phi codes, right? And the colors represent different, like, so first one is infectious disease, the blue, I guess this one. Second one is neoplasms. Third one is endocrine and metabolic, and so on and so forth. And here's the legend for that, right? I know, which is, yeah, it's circulatory system. No surprise, right? So, and the idea here is not to really dig into their findings, but I'm gonna talk about something that is interesting, I think. So this is a figure from that paper, right? And as far as I can remember, there are no MDs in this paper. They're all PhDs and data scientists and statisticians. And, you know, naturally they did their best to go after this important problem, timely problem, and applied a well-known tool to the problem at hand. But when it comes to interpretation, so you see this is basically, so if you focus on this part, right, deceased versus alive, and there are a bunch of things that are, crossing this significance threshold. And here's what's kind of bubbling up for the non-Hispanic whites, and here's what's bubbling up for non-Hispanic African Americans. So they put it in a Venn diagram. They said, okay, here's what's common to those two groups. Here's what's unique to non-Hispanic whites, and here's what's unique to non-Hispanic African Americans. And if you read the paper, it's something funny. Okay, so for non-Hispanic African Americans, circulatory system has like 16 things that are bubbling up, right? And that makes sense, right? It's COVID, it is attacking that system, it is, about that system, that's how we catch the disease, and the respiratory system, great. When you come to non-Hispanic whites, there's this dermatological findings, like 9 of them. So what is that? That doesn't make any sense, right? So you go like, what is collagen metabolism? Like, what is going on? You squint and you look at this, and you see it's decubitus ulcers, right? So bed sores. Well, then that makes sense, right? If you have that background, and if you can interpret these results, then you know that those patients don't really have a skin problem, right? Those patients were 14 days before catching COVID, bedridden, couldn't move, had decubitus ulcers, and were either suffering from some really bad chronic disease or stroke or something that really immobilized them. And these patients there's something else going on that resulted in that ICD-10 code in their medical record, and that's what we were able to detect. But you really need to have a team of experts that can interpret these results. Otherwise, these findings, associations, can be very misleading, and that's what I wanted to bring to your attention. So with that, I want to talk a little bit about a study that we do here. So I was lucky enough to get funded to work on this project, and it's about the PheWAS of polygenic risk scores. So polygenic risk scores are a combination of multiple SNPs that come together and create a single score for risk prediction. And we wanted to study 3 things. We wanted to study, first of all, the 2 polygenic risk scores that are developed by Dr. Hayek here at Stanford Imagenetics in coronary artery disease and breast cancer, and just replicate previous findings for individual SNPs, and then look at the pleiotropy of the polygenic risk score in the phenome-wide scan. And now we wanted to improve on that. But you can immediately appreciate that instead of a single SNP, just like the transcriptome or the copy number variations, one can take these polygenic scores that are combination effects of multiple SNPs and do a phenome-wide scan to detect these pleiotropic associations between various polygenic scores developed for a certain condition and the phenome-wide scan. So the 3 research aims is, 1, conduct this PheWAS using the PRS into phenotypes, and then improve the phenotyping by incorporating additional data other than ICD codes and NLP extracted concepts. And thirdly, instead of using ICD groupings or billing code groupings, create a human phenotype ontology-based encoder and grouping, which is our hypothesis is that it's going to be more representative of the actual phenotypic groupings that are observed in the EMR. So the first aim is that we will be taking the Sanford Biobank and Imagenetics combined data set with SNP data and linked EMR data that is approximately 30,000 subjects, extracting the phenotypes from the linked EMR, and then converting them to phe codes and running a PRS analysis, PRS-FEWAS analysis for these two phenotypes. Then, because we believe that the accurate identification of phenotypes is a critical factor in the power of FEWAS analyses and its ability to detect true associations, we will try to improve the existing phenotype encoding methods by incorporating laboratory, medication, procedure data, and the concepts extracted from the phe text. And then develop a method that can be used to do this in other similar EMR settings, which will be helpful to the scientific community. And lastly, again, to create, rather than a billing code-based groupings of these phenotypes, create an HPO-based grouping of these phenotype codes. So given that, so far, we've been able to get data for 25,639 individuals. Here's the female-male breakdown. Unfortunately, this isn't a randomized study, so it's basically voluntary enrollment into both the biobank and the Sanford Imagenetics program, and it's heavily skewed towards female individuals. But the age distribution, even though it's a little older than we would like, and you can imagine, prevention is a primary focus, especially in Imagenetics, and the younger a patient is, the bigger impact prevention would have in their health. But our patient population is approximately 60 years old in general, mean age. Then we took the 180 SNPs that are used to create the polygenic risk score. Then we checked the phenome-wide association against all those SNPs, right? So the phenome, in this case, resulted from the 417,881 unique patient ICD-10 codes. We then combined them into 1,855 phe codes, and then performed the FEWAS by controlling for age and sex. And here are some unpublished results. So there are positive controls, of course. It's good that we can detect the ischemic heart disease and coronary arteriosclerosis in this FEWAS study because these SNPs have been previously associated with CAD. That's why they're included in the polygenic risk score of CAD. But in addition to that, we are detecting additional phenotypes. Yes, lipid phenotypes, but Alzheimer's disease and dementias are the most significantly associated with odds ratios of 3 and 2.36. So this is really an interesting finding. It might be due to a shared biological mechanism. It might be due to a shared environmental impact. But this is the way we generate these hypotheses, and then we study these in the laboratory and independent cohorts and find an actual biology that can be targeted, hopefully, to treat patients more effectively. Here's the same thing in the Manhattan plot format. Here you can see that the phenome-wide significance threshold, corrected for multiple testing, is here, and then we have all the phenotypes that passed the significance threshold for these 180 SNPs. So each one of these, as you can see in this, can be associated with a different SNP from that polygenic risk score, but we have a total of these conditions that are associated with these collection of SNPs. Next steps would be to conduct the polygenic risk score analyses and then improve the phenotyping based on the goals that I alluded earlier as part of the goals of this research study. So with that, I would like to thank our patients and Sanford Biobank participants. Dr. Weimer, who is the PI of the grant, that under this research was done. Dr. Roder, who is a key collaborator from the Lundquist Institute and my mentor actually for this research project. Dr. Hayek, who is the clinical director of Stanford Imagenetics and is the PI of the research project that is developing these polygenetic risk scores. Imagenetic laboratories and staff, Lundquist Institute team, Stanford Health, EA, and Stanford Research Data Exchange Corps were instrumental in getting the medical record data for this study. And USC's Lawrence High Performance Computing Cluster and the team that supports it has been instrumental in allowing me to work with this big data problem. I thank you very much for your attention, and we're happy to answer any questions.
Any questions for either of our presenters, Ms. Fish or Dr. Sincan?
I'll jump in. Murat, Juliet, very nice presentation from all of you. So I'm wondering if there are any phenotypes in particular that this type of modeling or this type of analysis really has a hard time with. I'm thinking about things that are transient, iatrogenic diagnoses, things like that. I'm wondering how you deal with those sorts of items.
So FIWAS doesn't really, FIWAS is basically occurrence of a disease or a phenotype, right? And you can define your time frame. You can define and say things like, I'm only interested in phenotypes that persisted over time, so chronic conditions. Or you can say anything that was mentioned in the EMR, as long as it is mentioned, then I'm interested in looking at that. And some things, even if they're transient, as long as they're documented once, even if it lasted one day, that's still something that happened to the patient potentially. And it might have something to do with some genetic marker. And if it's an important health problem, then you might want to understand better the biology behind it. So the duration of the disease, unless you limit yourself to a certain duration when you're creating your phi codes, is not going to be an issue with this type of analysis.
I have a specific question and a general question then for both of you. The specific question, back up 2 slides. What were you testing for association within that FIWAS Manhattan plot there? The polygenic risk score?
No. So the polygenic risk score is constructed using 180 SNPs.
Yeah,
right?
in 25,000 people.
In 25,000 people.
Yeah.
So polygenic risk score is a formula that takes these 180 SNPs and weights them, right?
Yeah.
And in the end, you end up with a single number.
Yeah.
In this analysis, the single number is not used, but the individual SNPs that go into that calculation are used. So RS4803455 is one of those 180 SNPs,
And it's associated with Alzheimer's.
that is associated with Alzheimer's
So
disease.
show the Manhattan plot.
Sure.
So this is a Manhattan plot for one SNP tested against all those features.
So this is a Manhattan plot for 180 SNPs. So the x-axis is all the diseases, the 1,800
It doesn't
phenotypes.
require all 180 variants, just one of the 180.
Right, so
Gotcha.
this
I
one is an
apologize.
association between one of those variants and Alzheimer's disease. So when you combine them into a single number and run it, the PRS might not have this pleiotropic association. But the SNP actually does, and this is a novel finding.
So does effect size make a difference here, or just significance? In other words,
So this is
that
just
Alzheimer's
significance.
and dementia
This is just the p-value.
It's just very, very strong.
Yep. But the effect size is here as an odds ratio, and that's also significant, right?
Threefold increase in
Yeah.
risk. Gotcha. I have a general question too, unless you guys have more that you want to ask. Just see, you guys, this is really powerful, and you're finding all kinds of previously unrecognized associations in this retrospective or observational cohorts existing data.
Yep.
Right? Either one of you, any suggestions how you want to see this, or where you see it around the corner being implemented prospectively? Ice cream for drowning, that threefold risk SNP for dementia, something at the interface in maternal fetal medicine, if you're genotyping babies. Either one of you, something exciting around the corner where you think it's coming prospectively, or where you want to see it, because they're going to be driving the bus in the next couple of years. Anything prospective?
Comment? Prospectively, I think it's important to enroll a diverse group of individuals into studies like this, right? One thing that really impacts our ability to detect these things robustly is ascertainment bias. So when we have biobanks or clinical programs that are not necessarily recruiting a representative population to their database, that really impacts our ability to find these associations. Now, in this region, we don't really have a ton of diversity, but even within that population, we should be able to at least get a representative sample of our own population. I think that's really important in prospective studies. We can focus on that piece.
And because you pointed out, this cohort is a little older. The average age is 60, give or take.
Yeah.
And if you want to implement it in 1-year-olds or 10-year-olds or 25-year-olds, we need the width and breadth of the whole.
Yeah, and the older population is not necessarily something that's detrimental in the sense that you need to have time to accumulate these phenotypes, right? The EMR of an 18-year-old healthy male is really not that informative, if you will. But an elderly person has multiple conditions, then you have that information and you can detect the associations. However, if we don't have a representative, for example, sex-wise or ethnicity-wise, even if everybody is considered non-Hispanic white, ethnically, there are different ethnic groups, and we should still be able to get genetic data and phenotype data from all those different groups and not based on maybe one of those ethnic groups in this region. And also, there's a significant need for enrolling more, Native Americans that are prevalent in this region. So any type of prospective efforts should probably focus on those aspects.
This is population testing is what you're doing as opposed to specific disease.
Yeah.
I'm trying to get my head around DNA versus RNA testing in this situation. You're doing DNA testing.
Correct.
I mean, oncology has gone on to RNA testing and gone away from DNA testing. I'm just wondering about impact of RNA testing versus DNA testing.
That's a great point. I think oncology is really spearheading the application of genetics into clinical medicine, rightfully so. And yes, they are focusing on not only germline mutations, but somatic mutations at the DNA level, but also very relevant, temporally relevant changes in the expression levels by testing the RNA. So it's not just one thing. I think it's multi-omics approach, proteomics, metabolomics can also be used in these approaches. But taking the whole molecular profile, if you will, of the patient, and then comparing it against different phenome-wide scans will really improve our understanding of these conditions. It could be, we don't have to do a phenome-wide scan against the whole phenome. In this case, we can probably do a phenome-wide scan against the, neoplasms and oncology phenotypes.
Yeah, for RNA, you got to have tissue, Michael. That's why you guys are on point.
I'm thinking about association. Say you find out that there is an association for small cell carcinoma lung.
Yeah.
You find it in a 25-year-old, and you find the association that if you ever smoke, you're going to get it.
Sure.
Versus if you don't smoke, you won't get it. And small cell is basically curable.
Sure. So, just like any other basic finding, there's a gap between these types of findings, the association findings, and applying it to the treatment of a patient, right? This really helps us understand the relationship between small cell carcinoma of the lung and maybe kidney cell carcinoma, renal cell carcinoma of the kidney. But understanding those mechanisms helps us understand maybe there's a medication that we could, utilize in that population or will have an impact on that other disease. So that's more downstream, and unfortunately it doesn't have an immediate impact on the daily practice today.
I think we'll wrap it up then because the hour is long. Boy, I want to thank you both. Nice job.
Thank you very much. Thank you.